I met John and Jesse maybe about 16 years ago, something like that, and where they came to Ottawa and I think opened a lab dedicated to DMD research, and then worked with them for a while. But it's recently I was funded with a new grant from Jesse's Journey, so it's really a lot of fun to reconnect with Jesse's Journey and to see the amazing work they've been doing in the last 16 years. So thank you for having me. And let's get on with the show. Now, I'm a basic scientist. I'm trained as a molecular biologist. I'm a stem cell biologist. So I will try not to overload you with data, even though that's usually where I love to go. But don't worry, there's going to be an exam at the end of my talk. It's multiple choice though, so just pick B, you'll be fine. I would like to say that stem cell research has really paved the road for regenerative medicine to enter the clinic, and regenerative medicine is going to be a series of disruptive technologies that are going to transform the practice of medicine. And within our lifetimes, many of us believe we're going to see these new treatments coming into the clinic to treat genes, to treat diseases that before had no treatment at all or were not effective treatments. And I think this includes DMD. So I think there's great hope. We're really arriving at a tipping point where things are flipping over. We're seeing many new clinical trials occurring, whereas 16 years ago there was very, very little activity. So there's a lot of activity now and a lot of hope. By way of disclosure, the first half of my talk is funded by Jesse's Journey, and the second half is funded by, in part, by Satellos Bioscience. I'm a founder in Satellos Bioscience, so of course I'm totally biased, and you probably shouldn't believe a word of it. The muscle satellite cells are a subset of cells that live beneath the basal lamina as part of these muscle fibers. They make up about 3 to 5% of the nuclei beneath those myofibers. The rest are in the muscle fiber itself. Muscle fibers extend from joint to joint. Of course, they allow the muscle to move. There's a blow-up cartoon of a muscle satellite cell, and there's a real one stained with a protein we discovered called PAX7, transcription factor that controls the function of these cells. Now, it's a subset of satellite cells, about something less than 10%, that are the long-term self-renewing stem cell of muscle. The rest of them are more specialized and further down the path to making muscle and are not necessarily stem cells. It's the stem cell we're interested in. So satellite stem cells express our favorite transcription factor, PAX7. They can give rise to what are called progenitors or myoblasts. These are committed to only become muscle, and then these can differentiate to form these multinucleated myofibers. And if we do an acute injury of muscle with a snake venom called cardiotoxin, you can see we can blow it apart, and then muscle will regenerate. And muscle is incredibly regenerative. It will, if you are climbing mountains, lifting weights, it can regenerate. It's really very potent regenerative ability. Of course, this is perturbed in DMD, and I'll get to that. And so it's this population of cells that we're interested in targeting. It's important to know that stem cells can divide in different ways. A stem cell can divide and make 2 stem cells. This is called symmetric expansion, and it occurs parallel to the muscle fiber. It can also divide asymmetrically to give rise to 1 stem cell that remains behind, and then a cell committed to become a progenitor, myoblast, expand 8 to 12 times, giving you over 1,000 progenitors before differentiating. That's called an asymmetric division, and it occurs at a right angle to the muscle fiber. You can see pictures of these occurring here. There's an asymmetric division of a stem cell, leaving behind 1 stem cell and 1 committed cell, and here's a symmetric division of a stem cell. Now, we've learned over the last 15 years or so of research that the balance between symmetric cell division, making 2 stem cells, versus asymmetric division has a really significant impact on the repair process in a way that is intuitively not obvious. And that's because each asymmetric division makes this cohort of foot soldiers that affect the repair. So slight changes, slight perturbations in this balance can really have profound effects on the efficiency of repair. I'm going to be telling you about Wnt7a, which is a protein that's secreted, and I'll tell you about our work that's funded by Tessa's Journey in the next few slides. This is a protein that's normally made by muscle that stimulates the symmetric expansion of the stem cell, and this has a very positive effect in DMD. The problem is this protein is not very soluble. In aging, we also know that we show that there's a loss of symmetric expansion, loss of stem cells, and that using drugs or genetic means we can rejuvenate stem cells to be more youthful to give you the normal balance between symmetric and asymmetric division. Another way to think of this is a seesaw with symmetric division, asymmetric division. Normally there's a nice balance that mediates stem cell self-renewal and producing enough progenitors to affect maintenance and repair of muscle tissue. When we treat with Wnt7a, we slightly tilt it towards symmetric expansion, increasing the population altogether and allowing still for efficient progenitor expansion. And this really enhances muscle regeneration. And we've explored this as a possible treatment for DMD. In aging, of course, you lose symmetric expansion, you have more asymmetric division, leading to stem cell depletion and inefficient repair. And in DMD, we discovered a novel role for dystrophin in stem cells where we have loss of asymmetric division. We have too many stem cells that can't make enough progenitors, and this impairs the repair process. And we think this is a significant contributor to the disease pathology. We've shown, we've done a lot of work on Wnt7a, and we're continuing to develop it as a treatment for DMD. The problem with Wnt7a, which is it's a protein biologic, it's expressed by proteins and secreted outside where it signals to other cells, that as a purified protein, it's quite insoluble. If you inject it into the blood system, it just sticks everywhere and it doesn't go into muscle elsewhere. You have to inject it directly into muscle. So initially we were testing its activity that way by injecting it into a muscle. This is not an effective treatment for DMD because you can only treat one muscle at a time. So it might be help with grip strength or swallowing, that kind of thing, but it's not going to be a body-wide treatment. Our new work, I think, raises hope for developing a system to deliver it to muscles throughout the body. This just shows you that following acute injury, it's nicely expressed in muscle. There's Wnt7a making muscle, so it's an intrinsic mechanism for mediating repair. In at least MDX mice with DMD, they don't make it. So chronic injury doesn't result in Wnt7a expression. But we know that through many different studies that Wnt7a works at multiple levels to really enhance and stimulate the repair process. And we also show that in mice, it really has a remarkable effect. So this next phase of work is looking at how we can deliver this protein throughout the body. And it's been noted in recent research that Wnt, this family of proteins, as well as other sorts of secreted proteins, are secreted on small vesicles called exosomes. So they're not secreted as naked protein, they're secreted as these little particles, lipid particles, that then can travel throughout the body. And these are the mechanisms by which these particles are secreted and then taken up by a target cell. And so we're interested in asking, is Wnt7a similarly secreted on exosomes? And so what we did is an experiment where we damaged the mouse muscle, we isolated that muscle, then and minced it up and put it into a petri dish, and then we collected the medium, and then we purified the exosomes. And we developed a new method of purification that allows this to be done at scale. This could be done in a way that can manufacture very large amounts of this material. And in the red lines here, you can see we have very high levels of protein being expressed, and this markers here just prove that they are exosomes. So damaged muscle secretes a ton of exosomes, and these exosomes express Wnt7a at very high levels. And by electron microscopy stained with antibody, you see these black spots. They have very high levels of Win7a, so expressed at very high levels on these little vesicles. So the next question is does this have activity? And so we did a very simple assay where we're asking whether or not Win7a can stimulate growth of muscle fibers in a petri dish. This is a very quick assay for us to do. And the purified Win7a stimulates what's called hypertrophy, and so does the exosomal Win7a. But this is from the muscle explants, and the muscle explants contain all kinds of stuff. So we did this experiment again where we made use of a mouse where we could delete Win7a entirely, delete the gene. So this is a mouse without Win7a. And we made exosomes from mice expressing Win7a, you can see that in red, versus muscles with no Win7a, no red. And you can see on this plot here, here's Win7a, and there's no Win7a here. And we asked what would happen there. And indeed, the exosomes that contained Win7a had a very nice response, no Win7a, much less of a response. So these exosomes are very effective at transmitting the effect of Win7a. In fact, we need to have a lot less Win7a protein on those exosomes. So it's more effective than the purified protein. So we think this is the natural way of delivering Win7a. We know that vesicles can be delivered systemically, and we've done an experiment to begin to address that. So this is an imaging system, and we're looking at a cross-section of a mouse leg. This is a wild-type normal mouse without muscular dystrophy. And so this leg has been damaged, and we can see very nice uptake of exosomes, and exosomes that contain Win7a in particular, into this damaged muscle. So the next experiment is to deliver through the blood system by intravenous injection, just like it was done here, to an MDX mouse and ask whether these exosomes can mediate a body-wide therapy to enhance regeneration in mice with DMD. So, just speaking philosophically, there's multiple treatments now that are being explored for DMD. But I think many of us think that to reach a point where we really have an effective treatment, we're going to need a cocktail of approaches. We're going to need anti-fibrotic inflammation sorts of approaches. We're going to need perhaps gene correction and gene therapy for muscle fibers, and stem cell mobilization, and perhaps a few other things. Deflazacort may be part of that cocktail. But this is a difficult, complicated disease, and it's going to take multiple approaches deployed simultaneously to have the most effective, in my view, and the view of many scientists in the field, to really get at this as a treatment, an effective treatment.
Mike, do you see that cocktail having to be customized for each child?
That is possible, especially the genetic correction bit is going to be specialized. But I think these other things like anti-inflammatory, stem cell mobilization, etc., will not be. They'll be more generic. And those will become more important where there isn't a genetic correction thing available. Okay. So a number of years ago, now in 2015, we published a paper in Nature Medicine that was, I think, really paradigm-shifting discovery in DMD, and this discovery is slowly permeating the field, and so I think it's still worthwhile speaking about. The conventional view is that the disease is due to the loss of dystrophin in differentiated muscle fibers, leading to this loss of structural integrity, this breaks in membranes and ongoing damage and so on. We discovered a new role for dystrophin in stem cells that affects asymmetric division, and I'll explain it in a moment. And this impairs the regeneration of Duchenne's patients. We think that this is a really important contributor to the disease, and that it requires both parts, both roles to have the full-blown progressive disease. One, the ongoing damage in muscle fibers, combined with this reduced ability to repair that. And in support of that argument, the genetic experiments done in mice where the gene is deleted only in differentiated muscle has a very mild muscular dystrophy. If you delete the gene in both stem cells and the muscle fiber, you have the full-blown disease with the impaired repair. That's one piece of evidence. The second piece of evidence is the work from Lou Kunkel's lab with dogs, where they found an escaper dog who had the genetic deletion and should have the full-blown disease, but did not have it at all. That dog instead had a change in the expression of another gene called JAGID1 that stimulates stem cell repair. And that dog was perfectly normal and healthy. And thirdly, we've done experiments where we have stimulated and rescued this loss in mouse muscles, and those muscles have normal force generation and a normal histological appearance. So we think that the stem cell aspect is very important to pay attention to, and that correcting the deficit only in the differentiated muscle fiber is going to be insufficient to correct the disease. So this is a stem cell dividing asymmetrically. This is the basal lamina that wraps the muscle fiber. Stem cells align themselves between the basal lamina and the muscle fiber, so the muscle fiber here would be below. A stem cell, in order to divide asymmetrically, needs to know what's up and what's down, where the North Pole is and where the South Pole does. So what it does to recognize and orient itself is that it assembles dystrophin as part of this dystrophin-glycoprotein transmembrane complex at the basal lamina, marking that as the North Pole. Dystrophin recruits another protein called PAR1B to that pole. This is a kinase that inhibits the formation of the PAR complex, which instead forms at the South Pole. So now it's marked, the North Pole and the South Pole is different. It's able then to undergo a mitosis, or division into two cells with this 90-degree orientation, and give rise to one stem cell and one progenitor. In DMD, dystrophin is not there. That kinase is not recruited. This cell doesn't know where the North Pole or South Pole is. It's lost in space. It doesn't know where the floor is or where the roof is. And it tries to divide. Some cells do divide correctly, but others develop what's called multipolarity. They have multiple North Poles, and that cell arrests, undergoes what's called mitotic catastrophe, and is eliminated by programmed cell senescence. And as a result, we have this large reduction in asymmetric divisions. And this causes this imbalance where we have too many stem cells and not enough progenitors being made. So the flux from the stem cell compartment to the progenitor compartment is really, rather than being the tap being filled on, it's just a drip, drip, drip. And we believe this really significantly contributes to the progression of the disease. We just published a paper in January where we've identified a novel signaling pathway that rescues this loss of asymmetric division. And this is a complicated slide describing the signaling pathway, but it involves a protein, a transmembrane protein called EGFR that's located on the same side as dystrophin, and this works absolutely independently of dystrophin. And if we activate signaling through this receptor using a protein ligand called EGF, we can drive the same response and enforce polarity independent of dystrophin. And we've shown that using expression vectors where we drive EGF in individual muscles, that these muscles are no longer dystrophic. They generate force levels that are the same as a wild-type muscle. And this is strictly due to stimulating asymmetric division to normal levels, generating normal levels of progenitors, and just driving the repair process full on. And so a very effective way, simply by targeting a stem cell without correcting dystrophin, to enhance the repair process and restore functionality to the muscle. So how do we do this across the whole animal? We don't want to turn EGFR on where it's not supposed to be turned on. We don't want to use EGF, for example, to drive this or other mechanisms to do this. EGFR is a touchy molecule. It's also involved with breast cancer and other sorts of things. We have to do this in a very context-specific way. We have identified a target. This is shown here as PTPX, which normal role is to turn off this pathway. It's a negative regulator of this pathway. And when this pathway is activated, what happens normally, it comes on and it's immediately shut down by this protein. This protein is very highly expressed in muscle stem cells, and what we found is that if we block the activity of this protein, this signaling pathway is maintained at normal levels. So it still needs to be turned on normally, so it's context specific, so the muscle damage turns it on, but rather than being turned off right away, it's being maintained at high level expression. And we've been developing drugs that inhibit this protein to keep this pathway on. And these drugs are very effective. So this just shows you a figure of work from our paper that we published using EGF. And you can see that the muscles are larger. These different colors are the muscle fibers. So we're looking, it's like cutting a dowel, and you're looking at the end of the dowel, so it's like a little circle. You're not seeing the muscle fiber. And you can see that these are bigger, they have bigger mass, and they generate force very effectively. So they're stronger than untreated muscles. And if we look using this time a drug, our first generation tool compound, again we have a very dramatic effect simply by stimulating muscle repair. So these white fibers are dead fibers. These little green ones are newly regenerating fibers trying to repair that damage. And these red fibers are the more mature fibers that are being lost. If we treat with drug, we have much reduced fibrosis. We don't see these focal areas of dead fibers and regeneration. We just have this histologically much better appearing muscle. And of course, we've done all the measurements that go along with that. But this particular compound we can't use clinically. I'm out of time. Or are you just waiting? Okay. All right. So together with this company, which I'm a founder in, don't believe anything I say, we've been going through a medicinal chemistry program to make a specific drug that can be used in the clinic. And we've made amazing progress in the last year and a half. So using a variety of different approaches, we came up with 5 families of compounds, and we've selected one lead that we're working on now. We understand what needs to be done, and we hope by January to have our clinical lead in place. And this is really an exciting development, a totally novel way of treating this devastating disease simply by stimulating the repair process. Now, will this be used alone? There's a lot of growing to be done from being an infant to a 20-year-old, and we think that there'll be that if this is effective in humans, there's a lot of work to be done. That this is something that would really help keep the pace of growth and allow regeneration to occur from just normal wear and tear, as well as from addressing the problems from the disease. But where it is especially attractive is using it in conjunction with gene therapy. And I think that's a really interesting and exciting application. The gene therapy vehicles that are being used using adeno-associated virus only target muscle stem cells very poorly, and the ones that have been corrected aren't maintained. The virus itself doesn't integrate into DNA, so it'll be diluted out, so it will not be useful in the long term. And even when using CRISPR-Cas genetic correction, it's diluted out quite rapidly. And that's because we think that the mutant stem cells have a expansion advantage, and they take over. So this kind of approach, we think, will be very important to restore the balance of stem cell self-renewal and commitment in DMD, and has really exciting potential for therapeutic applications. Next year, we start the IND enabling studies. Those are studies that allow an application to the regulators to do a clinical trial, and we're really hopeful that this could be initiated within 2 years. And these are the people who've done the work. Thank you very much.